Hi, everybody, and welcome back to Biology Bios. My name is Jim Edelman. I'm an associate professor in the Department of Biological Sciences at the University of Memphis. And my purpose with this podcast is really twofold. First, I want to let folks know that most Thursdays during the semester, we have talks by visiting researchers at 4 p.m. in Ellington Hall. And that's folks from all over the country and world talking about the really cool biological research that they do. The second purpose is to have those folks here learn a little bit more about them, how they got where they are, and what really excites them about the research they do. In addition, I'd like to say a quick disclaimer that I borrowed these questions from Rhett Miller's excellent podcast called Wheels Off, so please check that out as well. But without further ado, let's get to this week's guest. Thanks so much for being here. Dr. Joe Opferman, thanks so much for being here and welcome to Biology Bios.
My pleasure. Thank you.
So generally I have folks just give a really brief intro to who they are, what they do, kind of your elevator pitch sort of length, the broadest brushstrokes of where you are, what you do.
Okay. Yes. So I am a researcher at St. Jude's here in Memphis. And my laboratory is really interested in trying to understand the regulation of cell death. And part of the real excitement about what we're working on now is trying to understand how the cell death pathway integrates with other pathways in the cell. So the cell death pathway basically controls the lifespan of a given cell. And so this is a genetically programmed system that controls how long a cell should live and die. And it is regulated by a series of proteins called the BCL2 family of proteins. And this is made up of both pro-apoptotic, so genes that kill cells, and genes that protect cells, so-called anti-apoptotic genes. And it's the interplay between these proteins that basically regulates the cell survival or death. And so, of course, this is very interesting to both normal researchers because it's important for controlling the survival of millions of cells in your body every day that go through this normal turnover process. But it's also incredibly important for cancer biology because cancer cells often violate checkpoints that should make them die. And this is one of the main pathways by which these cancer cells are their survival is promoted in humans and in models as well. So we're very interested in trying to understand that regulation of how this process is controlled. And we're also very interested in trying to understand how it may impinge on other normal functions of the cell, such as the cell's metabolism.
All right. Yeah, very, very cool. I often think about apoptosis from an immune standpoint, right? From your killer T cells promoting apoptosis when they find a infected cell. So
that's actually how I got my start.
Oh, really?
I got my PhD in immunology and I worked on cytotoxic T cells.
Yeah, you know what? Now that you mentioned, I was like, yeah, I remember that from internet stalking you a little bit ago.
Yeah. So that was actually one of the things that really got me interested in this process. So there are two really cool things about the immune system that really drove me into this field. One of which is during the immune response, you normally go through this tremendous expansion of cells. So, a few cells that are specific for a given antigen will expand literally thousands to millions of times. And those cells accumulate in the host, they clear the pathogen, and then they have to be eliminated. Otherwise, by the end of our lifetime, we'd be just one big lymphocyte. And so that process is really one of the things that I found so amazing about the immune system. And that process is fundamentally regulated that's an apoptotic process by which these cells are dying. And then furthermore, as you pointed out, cytotoxic T cells, when they engage an antigen presenting cell, they actually deliver a lethal blow through granzymes and perforin. And those granzymes actually activate the apoptotic pathway. And that's also very important for CAR T cell therapy, which is being used in immunotherapy right now. So we're trying to understand exactly how cancer cells, which they're often quite sensitive to antagonists of the BCL2 family, are they more susceptible to CAR T cell induced killing as well? So there's lots of really cool aspects that the immune system brings to apoptosis research.
Yeah, absolutely. Okay, so sort of, I guess this kind of Well, I was going to say maybe this does, maybe this doesn't merge those two areas, but you've talked a lot so far about like these interactions with apoptotic pathways, and I'm wondering, is there like a current project in your lab, maybe that does that sort of thing, that looks at this crosstalk?
Exactly.
That
Yeah.
is there a project you're particularly excited about right now?
Yeah, so there are, we have a lot of really exciting projects in the lab, but one that I think is, that I will highlight is a recent publication that we had in the journal Molecular Cell, which basically showed for the first time that that one particular anti-apoptotic protein called MCL1, that it actually not only promotes cell survival, but it also is required to facilitate long chain fatty acid oxidation by the mitochondria. So this
Oh,
is really an amazing and novel identification of a regulatory pathway by which two things that happen at the mitochondria, cell death and also beta oxidation, are both controlled by the same enzyme. And so, but that's actually not an enzyme. It's controlled by a protein that regulates the enzymatic process of fatty acid oxidation. And so this is a really amazing discovery in my mind because it explains some data that had been in the literature but not very well understood that argued that loss of MCL1 may affect fatty acid oxidation. So now we've mapped that molecular pathway. But it also explains one of the curiosities that has been observed in clinical trials of MCL1 inhibitors, which are being developed for lots of cancer therapeutic strategies. These MCL1 inhibitors in clinical trials have often been associated with cardiac damage. And that cardiac damage has actually been a
interesting.
real limit on their ability to be approved by the FDA. And it's actually caused a lot of consternation. And so it's been hard to understand exactly why this pro survival molecule is causing heart damage because typically your heart cells are very resistant to apoptosis naturally because they don't recover after damage. So you want them to be very resistant to any
Right.
sorts of apoptotic stress. So it's very surprising to many people that MCL1 was required for this stage of cardiac survival. So what we think is going on is that this role in fatty acid oxidation by MCL1 is also very important for cardiac biology, and that's because cardiomyocytes derive about 60 to 90% of their ATP from fatty acid oxidation. So if you
Okay.
are inhibiting fatty acid oxidation, which we showed in our mouse model we were by MCL1 inhibitors, we were able to demonstrate from that we could actually repress fatty acid oxidation. And we hypothesized that repression of fatty acid oxidation could also be contributing to this cardiac damage signature that they're observing in humans that are in these phase one clinical trials. So I think that this
Wow.
is an amazing discovery that may explain some of the toxicities of developing inhibitors, but it also has a fundamental biological mechanism. It's really striking that you would use the same protein to both regulate the survival, but also to facilitate energy production by these mitochondria. So I think that's an amazing discovery.
Um, I'm assuming, but maybe wrongly so, that this is a nuclear encoded gene, not a mitochondrial encoded gene.
No, you're correct. It is a nuclear encoded gene.
So yeah, I do. I think there's a lot of like, even from an evolutionary, like Evo Devo even standpoint, like that interaction between the nuclear DNA or nuclear encoded genes and this really important mitochondrial function, that's super cool.
Yeah, but that's so common throughout biology
Yeah.
because remember the mitochondrial genome only encodes for 13 polypeptides, and mitochondria are made up of literally thousands, if not more than 1,000 proteins. So many proteins from the nucleus are being brought into the mitochondria, and all of the critical work of the mitochondria is really dependent upon these nuclear encoded genes. So I think it is amazing that this actually it's a very interesting developmental question as well because the way that apoptosis genes were discovered was originally in the C. elegans, the nematode, and those family members are, of course, as you go back in evolution, there's only a single family member that plays the role of the anti-apoptotic gene, and that molecule in the C. elegans actually looks has some homology to MCL1 and to other anti-apoptotic proteins like BCL2, but it's actually not an MCL1 homolog.
Hmm.
So MCL1 is actually quite distinct from that earlier primordial molecule. In fact, about 50% of MCL1 is an intrinsically disordered protein on the amino terminus that is not even shared at all with that C. elegans ortholog. So that suggests to us that there's some sort of developmental pathway that has been achieved as we went through development and got into higher species. So I think it goes back as far as like zebrafish and I think Xenopus also have an MCL1 homolog, but it's not found in the lower species.
That's interesting. So it's not a
Yeah.
simple gene duplication. It's something else.
Well, it's probably is partly a gene duplication, but it's different. It's because it's twice as big as the other family members. It's sort of gotten another function that is still unclear. And because it doesn't have any like structure, it's really hard to identify exactly what that comes from. So sequence wise, as a primary amino acid sequence, it doesn't have homology to really anything. The question is, how does it fold? And is that folding influenced by other proteins that it interacts with? And I think there's a lot to still be discovered in that world.
Yeah, absolutely. Yeah, you got your hands full there, I think.
Yeah.
Okay, so I want to shift a little bit and talk about like your path a little bit more. So like, was there a time when you knew that you wanted to be a scientist, or was this a sort of slow discovery? What led you down this road?
Yeah, so I would say I was always intrigued with science all throughout my childhood. My grandfather was a pharmacist, and I always thought that was an amazing line of work, and I was interested in it. But no one else in my family was a scientist. No one else in my family was in the medical profession. But I always was drawn to science, and I mean, maybe I had an affinity for it as well. And I enjoyed it. But I have to say, when I went to my undergrad, I didn't sign up to be a scientist initially. I didn't think I wanted to be a scientist. At first, I thought I would want to be maybe an engineer. And then I thought maybe I would want to go into business like my father. But I always was sort of protecting that pathway. So I decided when I went as a freshman, instead of taking the typical science for business majors, I took science for science majors. And I really I never looked back. I immediately, so we didn't have to declare majors as freshmen. We had to declare them after the end of our freshman year.
Mm-hmm.
So I never declared as any other major, but I never went into anything other than science. So it was kind of like, I don't know if I always knew that was where I was going to go, but I always protected that path because I knew it interested me. And I think that's an important part of like what you do for your life. It should interest you. And if it doesn't interest you, it's probably not the right path. So for me, that was the path that I loved. And then I have to say, as I went through I was a biology major. So that means I wasn't a biochemistry or molecular biology or bioengineering. I took classes in ecology. I learned about moose and wolf predation. I learned about, I went out and measured the size of trees and tried to understand the density and the size of trees in the forest. But I also took a lot of cell biology and molecular biology classes and genetics classes. And it was really my senior year, I took a graduate level class in immunology that really was the thing that made me know that that's what I wanted to do. And what I loved about it was I learned about this sort of common evolution that we as humans go through, where we're exposed to all these pathogens around the world, and how we've evolved ways to fight off those, and how the pathogens also change the way they attack us. And it's this constant evolution. And I found that was just so amazing and so cool. And so you can learn so much about the biology of the immune system by understanding the pathogen. So I thought I wanted to go into infectious diseases. So I entered a PhD program in immunology. And I ended up working on more basic aspects of immunology. I worked on I looked at thymic development, and then I started to work more with cytotoxic T cells and trying to understand where they came from, what was their origin, what were the signals that were needed to keep them alive. And then that's sort of what drove me to this apoptosis, as I shared with you earlier. But yeah,
Yeah.
I wasn't always sure, but I think I always had a sense that I wanted to be a scientist.
Well, I like the idea too of protecting a potential path. That like, and I think that, and we're going to kind of talk more about like advice for students coming up now, but I think as we're younger, we often think I've got to do this one thing. I've got to go down this road. But like you were saying, you can keep a lot of doors open.
Yeah, I
And
agree.
and yeah, I
I
think
mean,
that's
that's
really cool.
advice I always give. I mean, I gave it to my own son who's in college now, and I've given it to lots of people over time. It's like, you know, you don't want to restrict your path because you decided at one point that you thought that you would never want to do something. That's I always wanted to be opening doors rather than closing them. But it doesn't mean that you can't go back. I mean, my wife is a prime example of a person who thought she wanted to be an economics major, and she ended up in graduate school too. So you can take either path. It just may take you a little bit. Maybe you have to take a few more classes over the
Yeah,
time. Yeah.
yeah, yeah, absolutely. Okay, so a bit of a gear shift here, but something that I think comes up a lot for people, graduate students, undergraduates, I mean, everybody, I think, in academia at some point, is that we have an inner critic that, will tell us, hey, that's not a good experiment, or hey, you don't know what you're doing here. And I'm wondering how you deal with that inner critic, how you either silence it or, I've talked to some folks that can make it work for them in some ways.
Yeah, I mean, I think it's good to be your biggest critic. I mean, I've always used that as my mantra. I don't want to be told that I'm not, that something isn't right. I want to be the one that knows it's not right. So I think that that's always been my sort of mantra to always want to be my biggest critic. But it can be hard too, because then you don't let yourself get away with anything. And sometimes you hold yourself to even higher standards than anyone else would hold you, and that's sometimes hard to deal with as a day-to-day life, because you know science, especially research science, is basically, it's you're trying to discover something new, and by it being new, that means you don't know what it is. So you may make wrong steps along the way, and you may never know enough. You may always be craving for more, and if you can't be happy with the discoveries you've made, sometimes it can be a hard existence. You have to be able to convince yourself that something is true, but there has to be a time where you wrestle with understanding that that's the best you can do with that topic. You need to document that, and then start asking the next questions. And that's the thing. Like, every time we write a paper or a grant, you're trying to make that story as tight as possible. And biology can sometimes be a little gray. It's
Oh yeah.
not as black and white as some of the physics or mathematics or things. And that's something, when you have it when you're innerly critical. Sometimes it takes a higher burden of proof to convince yourself that even your own work is correct. Now, I think that's an important step, though, in science, because the fact of the matter is that your name, your reputation is staked upon the validity of what you do. So if you don't have that inner critic, and you are more accepting of less proof, you can make missteps that could come back to haunt you. And I think that that's a big problem in the world of science overall right now. I mean, we often hear in the news about how many scientific things can't be reproduced, and I
Right.
think that that's always a danger. So I think having that inner critical voice is critical to the success of your program, and also the long-standing nature of it. That people are always going to, they're going to trust what you say, and they're going to be able to reproduce what you do. And so I think that's, like, the graduate students in my laboratory that I've trained. I think that we have to always be asking that extra question. How can we show this yet another way to make it even more compelling? but it's taxing.
Yeah, I think you hit on something that like there's a lot of value to this. And I think that you need to be, it sounds like what you've been able to do is be mindful of the value and be like, okay, so this inner critic is telling me, okay, let's think hard about this, which is what we should be doing as scientists, right? We should be thinking hard.
That's right.
And that's, I think that's a really good point about it that I don't think anybody's brought up that like is kind of integral into the scientific process. And yeah, I mean, as I feel like, at least for me, you have to have that balance, like you were talking about, like that sometimes it can paralyze you, right? Like then you can't move forward.
That's
Uh,
true. I mean, and like as when you have things like papers which don't have, when you're writing a manuscript, there may not be a specific deadline. That inner critic can delay
mm,
those things
yeah,
because you may never reach you may be much harder to reach that inner threshold for the rigor that you expect. But when you have deadlines for things like a grant submission,
yeah,
you have that grant and it's due tomorrow, you have to make a call like, do I think that does this meet my own personal level of what I think is right? And and what are the other things I would like to have that I don't have right now, and how can I like make myself feel okay without those extra pieces of data? So
yeah,
those are the struggles of the inner critic.
yeah, that makes sense. So you kind of touched on talking and training the graduate students in your lab. Um, but I was wondering in particular, and actually this might pertain to, I would normally ask this about undergrads, but this I think would pertain to graduate students as well. But what would you give like say late undergrad, early grad version of yourself, if you met that person today in today's world of biology? What advice would you give that person?
Oh, that's a great question. So the piece of advice I still give all of my students, and and I think people also gave me this advice, and I would have given it to myself as well, is you need to see lots of different things in science. Like the more like types of research that you're exposed to, the better off and the better rounded you'll be. So that
Mm,
means not just like one technique, that means not just one cell type, that means maybe not even just one system, but the more exposure, the broader your exposure to different aspects of biology, the better off your training will be. And I think that's especially important in today's research because it's very hard to have research that is single-minded in one particular technique or one particular aspect.
sure.
The breadth of that knowledge, the impact to broader fields makes your work so much more important. And there's so many new techniques that you have to be able to marshal those to really make your point in the most clear and the most impactful way. And so, like I always tell people, like, it's hard sometimes when you're in a city like Memphis because people come here, they love it, they want to stay here, but that we don't have tons and tons of different opportunities for different research. There's not lots of biotech here that they can be exposed to. So I always try to push people like you should go when you're done here, you should go see some other way of doing it. And I made that choice. Like
Yeah.
I went from University of Chicago where I did my PhD. I went all the way to Massachusetts to Dana-Farber Cancer Institute, Harvard Medical School, and I learned so much. I learned so much about like what it took to be successful. I learned so much about different aspects of science. I learned about different techniques, things that I never would have done as a graduate student. I got exposed to whole new areas. I went to seminars by people on topics that were nothing related to what I worked on, but just to understand what they were working on and to hear how they did it. And that exposure just gives you a much richer set of knowledge, and it also can transform your science and make you much more impactful.
Yeah. I mean, even if you get one nugget out of not even just every seminar, but even 1 in 10 seminars that you go to, you get one nugget that you can apply to what you've been doing. That can be transformative.
Yeah, for sure. I mean, especially as fast as technology is moving forward for like things like super resolution microscopy or things like that, which didn't even exist 10 years ago, that we had no access to them, to be able to see people present that work and understand like, oh my gosh, that would be so cool if we could do that. And to be able to utilize those types of ideas for your own research is really valuable. So I
I
think that
think,
that's probably the biggest piece of advice I would give is, get outside of your own lab,
yeah.
go see lots of different things, go hear lots of different talks and different subjects, and also transport yourself to a whole nother place, because you're going to see a whole different perspective.
Even just the people too, right? Because I mean,
Yeah.
we're scientists are people. So
Yeah.
if you do your PhD at a different institution than you did undergrad, it's a whole different set of professors. It's a whole different set of postdocs. There's
Yeah.
all those different perspectives.
Yeah, and I mean, and there's differences in so many different things about about the some places you're going to have like these hyperactive labs where they're working 24 hours a day. Just to see that with your own eyes, it's like is amazing. And then you come to a place where like where people work more of a 9 to 5 job, and it's interesting to see all those perspectives, and it helps you to decide what you want to do with your life. Where do you want to find, what's your niche that you
Yeah.
can fit in?
Yeah.
Now, the one place I never did try to go to was biotechnology, but clearly that's a whole area of research that I would have probably, would be interesting to be exposed to that and see the differences there. Like pharma or biotech work, but you can't do everything. That's the other
No.
piece
Yeah.
of advice I would give myself is you can't do everything. Like no matter how hard you try, you eventually have to focus.
Yeah. I mean, you can still keep those doors open, or maybe they become windows, right? Like maybe you
Right.
glance through to biotechnology every once in a while, even though that's not what you do with your day to day, right?
That's true. That's true. And the other cool thing is you can be exposed to it even through collaborations as a basic
Hmm.
scientist in academia. Sometimes those people will reach out to you for ideas or even to share reagents. So you can
Yeah.
sometimes get a window into their world.
Yeah, absolutely. All right. So a quick gear shift here. What do you do when you're not doing science?
So I'm a proud father of 2 boys, one of which is in college. The other is still in 8th grade. And so we do lots of stuff as a family. So I have been active in Boy
Hmm.
Scouting, Scouts of America, for a long time. My older son is an Eagle Scout. My younger
Cool.
son is first class right now. We're actually planning our first backpacking trip to Philmont in New Mexico
Oh, yeah,
this
I've never
next
been there, but
summer.
it looks amazing.
Yeah, so that's one of the things I'm going to be doing, is doing a lot of practice hikes in the area. But I do love to do. I love to go hiking. I mean, we love to go to Shelby Forest and places like that in Memphis
Hmm.
that are really beautiful, close outdoors areas. I love to play frisbee golf, so
Nice.
that's one of the hobbies that I picked up during the pandemic that I thought is something that I've stuck with. I also like to play real golf too. I'm not very good at it, but I like to play with my younger son is actually quite a good golfer and probably could almost beat me at this point, which is depressing for me. But I don't take myself too seriously when it comes to that. I also love to go fishing. I love trout fishing. I don't do as much of it as I'd like to, I must say. Maybe as I get more free time in my life, I'll take advantage of that more. I also love to cook and I like to read, and I just enjoy my life.
Yeah. Yeah. I
Yeah,
mean, sounds like you don't have a whole lot of time for trout fishing right now.
yeah. Sadly, it's the type of thing that it's wonderful to get with it when you can. Um, I just haven't made it a priority. That's the reality. It takes being a priority to
Sure.
really do it well.
Sure. Um, well, I have one more question for you, which is going to flip this totally back onto you and ask you, what else do you wish that I asked you about that I didn't ask you about?
Oh, um, it's a good question. So, you might ask how I ended up in Memphis.
Hey,
How
yeah,
I ended up
absolutely.
in Memphis when I was at the Dana Farber Cancer Institute, and I was grew up in Minnesota, and I did all of my training within 5 miles of I-90. So how did I get to Memphis, to St. Jude's
Well,
Children's
you probably
Research
took
Hospital?
57 at some point, right? No, I'm just kidding.
Oh, yes, many times. So many times, I think I know every restaurant and rest stop on that road. In fact, my parents still live in northern Indiana, so that is my route
Okay.
home.
Yeah.
But so what happened? The reason I ended up in Memphis was, it was a little bit of a fortuitous aspect. So I was working on a particular molecule that some investigators at St. Jude were interested in. They thought that they had identified a critical pathway, and they wanted some reagents from me. And so as I shared some reagents with them, I was just a postdoc, kind of like minding my own business in Boston. And they invited me down for a seminar, which was, I just thought I was coming down here to give a talk about my work. And then they were like, hey, what would it take to bring you to Memphis?
Oh, wow.
So I was sort of like, slack jawed and trying to figure out what I was going to do and trying to explain to my wife, like, are you interested in moving to Memphis? And she was working in the pharmaceutical industry in Boston. So it was a little bit of a culture shock, but, we came down and we came down as a couple and looked at Memphis, and we really fell in love with it. And now 20 years later, we're still here to this day. So sometimes a surprise like that you're not expecting can really be life-changing.
And I think that also speaks to, it kind of comes back to that openness of like seeking out new experiences.
Yeah.
When somebody calls you up and says, hey, we're working on this thing. Do you have this, I don't know, antibody or whatever it was? Like, yeah, go ahead. Strike up that collaboration, like make that connection.
And it worked out really well. It worked out great for me. And both my boys were born here in Memphis. So, I've lived now longer in Memphis than I did in Minnesota. So, that's quite a statement.
Yeah. Yeah. Well, thank you so much for all your answers, and for taking the time here today. Definitely looking forward to your talk next week. And, really appreciate you being here.
Great. Thank you.
Thanks again to our guest, Dr. Joe Opferman. You can check out his talk on Thursday, November 21st at 4 p.m. in Ellington Hall. Looking forward to seeing you there, and thanks again for tuning in.